Hello, everyone. Welcome back to my video tutorial for the Maxfuse package. In my previous video, we created 4 matrix data for the console CODEX and RNA-seq data. So today we can use the 4 dataset to perform Maxfuse analysis. In the first step, we create a fuse object. This object is the main object for running Maxfuse pipeline. We can use the function called model fuser to create the object. You can see we can set the arguments for shared array 1 as RNA shared, shared array 2 as protein shared. Both shared arrays will be used to find the initial pivots. We set the active array 1 as RNA active and the active array 2 as protein active. Both active arrays will be used to refine the initial pivots. For the labels, you can just set the none. If you set the none, Maxfuse will run the clustering to fill the labels. So let's create the object now. Then for the Maxfuse analysis, we split the fuse object into batches. This will reduce the computational complexity for the analysis. There are some arguments you can set. The maximum outward size will be the sample size for the smallest one. In our case here, will be the RNA sequencing data because the protein data has much more cells. And the matching ratio will be the protein sample size against the RNA sample size. You can set the meta cell size here. We set as 2. So now we can split the object. So we created the fuse object using the 4 matrix data, and also we split the object for the Maxfuse analysis. So next step will be constructing the UMAP graphs for 2 active arrays. So we can have a look at the Nature Biotechnology paper again. You can see in the pipeline, in the first stage, we used the 4 matrix data and created a fuse object. So next step, we will construct the nearest neighbor graph for both active arrays. So that's the second step we are analyzing at the moment. So to construct UMAP graphs for 2 active arrays, first we need to find the best principal component in order to construct the best nearest neighbor graphs for each modality. So we can plot the top singular values for active array 1. We can set the target as active array 1. So active array 1 will be the active RNA-seq data. So let's plot the singular values for the active RNA-seq data. Okay, you can see we generated the figure. This figure is very similar if you perform the elbow plot for Surat package to identify the best PCs for PCA analysis. You can see the elbow shape. Sometimes there are comments about this elbow shape. How can we determine the best PCs? So if you can see from this elbow shape, we can see that here is the elbow. So normally you don't choose PC value in this region because the PC value in this region cannot capture the full information. This is the elbow shape. If you choose a value on the forearm, then you will be okay to do the analysis. For example, here the online tutorial choose the 40 pieces for the RNA active data to perform the nearest neighborhood analysis. So we will follow the online tutorial, use the 40 pieces for the RNA active data. Next, we can plot the singular value for the active array 2. We know active array 2 will be the protein data. Let's run. You can see we generated the figure. You can see the elbow shape. The online tutorial used the 15 pieces. I can see if you use PC after 10, any PC will be fine. So we identified the best PCs for both active array. So now we can use the construct graphs function to construct the nearest neighbor graphs for both active arrays. So for the components 1, we set as 40. Then component 2, the protein data will be 15. We set neighbors for both data as 15, resolution as 2. Then we can run the construct graphs function to construct the nearest neighbor graphs for both active data. Let's run. So you can see we are analyzing the array 1 first. We aggregate the cells into meta cells at the average size 2 because we set the meta cells size as 2. In array 1, we only have one batch 0. You can see graph clustering finished for array 1. Now it is analyzing the array 2. In array 2, we have 5 batches, 0 to 4. You can see from the analysis in array 1, it constructed the graphs, then clustered the graphs as well. So now we finish the construction of nearest neighborhood for both active arrays. So in next step, step 3, we can find the initial pivots using two shared arrays. We can have a look at the Nature Biotechnology paper pipeline again. You can see we constructed the nearest neighborhood graphs for both active array. So next we can analyze both shared arrays to find the initial pivot. On the figure you can see here, after this step analysis, we will get the matched pairs for stage 2 analysis. So now we can run the analysis for the third step. Again, we need to find the best pieces for both shared array. So first we can run the singular values plot again for the shared array 1. This will be the shared RNA data. Let's run. You can see we generated the figure. The online tutorial used the 25 pieces, so we can just follow the online tutorial. So next we can plot the singular values for the shared protein data. Let's run. You can see from the figure, the online tutorial choose the 20 pieces for the protein shared data. I would say you can use any PC after PC8 for the analysis. So now we can use the function find the initial pivots to compute the initial set of matched pairs. So we can set the components 1 as PC25 for the RNA shared data and components 2 as 20 for the protein shared data. So in this function, you will see the arguments WT1 and WT2. So you need to set a value for the WT1 and the WT2. WT1 is for the component 1 dataset and the 2 is for the component data 2. So this value will be between 0 to 1 because Maxfuse use a method called fuzzy smoothing to find the initial matched pairs. So the smaller this value it is, then the greater the strength of fuzzy smoothing. So the highest value is 1. If you set this value as 1, then there's no smoothing method applied for the analysis at all. So here, the online tutorial suggests us use 0.3 for both datasets because the linkage between CODEX data and the RNA data is very weak. So the analysis requires stronger fuzzy smoothing. So now we can run the analysis to find the initial set of matched pairs. We can run. Okay, it is done. So we find the initial matched pairs for both shared data. So we can have a look at the analysis pipeline on the Nature Biotechnology figure. You can see we computed the nearest neighbor graphs for both active array. Then we find the initial matched pairs. The matched pairs is for the shared data because they have the same gene names in the columns. Because the weak link between the shared data, if we create a joined matched pairs graph, then the graph will be not in the best integration between protein and RNA data. We clustered the graphs for the active array because the active array contains all the features. So the graphs in the active array will be the best clustering. So now we can use the information in the active array to refine the graphs for the shared array because the gene names come from the active arrays. So in this stage of analysis, there's a function in Maxfuse called refine pivots. It will repeat this process until we get the best pivot matching. So now we can go back to Python again and run the stage 2 analysis. So here I didn't label stage 2, so it will be step 4. We run the analysis to refine the initial pivots. So in this step, Maxfuse uses canonical correlation analysis. So first we need to find the best number for the CCA analysis, so we can plot the canonical correlations. Here we set the first component as 50 and the second component as 9, then CCA component as 45. Let's run this code to create the plot. Okay, we generated the plot. So we can have a look at the online tutorial. The online tutorial used the 25 as the best CCA component. We can follow the online tutorial use 25, but I think any number after 25, you will be fine to do the analysis. So we can use the refine pivots function to refine the matched pairs. So you can see we use the same value 0.3 for the analysis, and the component 1 is 40. That's the value we choose for the RNA active data analysis, and the CCA component will be 25. So here we set the n iterations as 1. So this value is set between 1 to 5 because of the weak link between CODEX and RNA-seq data. So we use lower value 1. If you increase the value, it will degrade the performance. So let's run the refine pivots function to refine the matched pairs. So we refined the pivots for the matched pairs. So because the weak link between the CODEX and the RNA data, so not all the matches are in good conditions. So we can filter away the unreliable pivots. This step analysis could help later when we do the propagation step analysis. So here you can see we can use the filter bad matches function. The target will be pivots. So here we use the filter proportion as 0.5. The filter proportion specifies the proportions of pivots to be filtered away. So here 0.5 will be 50%. If you look at another example for the CIT-seq analysis, they use the 0.3 to filter away the unreliable pivots because in that dataset, the protein data has 228 proteins, so the link is stronger than this one. So here we set the filter prop as 0.5. Then we can filter away unreliable pivots. Let's run. Okay, the bad matches should be filtered away. So after this step analysis, you can see the note here. If you use the Maxfuse perform other analysis, for example, the CIT-seq with RNA data or RNA-seq with ATAC-seq data, because the link is strong, then you can start the co-embedding for full dataset into CCA space. But for the CODEX data, because the link is weak, so we run the propagation step before we co-embed the full dataset into CCA space. So next step, in step 5, we run the propagation analysis. For this step analysis, we can use the propagate function in the Maxfuse package. If we have a look at the pipeline in the Nature Biotechnology paper, you can see now we are in stage 3. Because we refined the pivot matching, now we run this step analysis to propagate the matched pairs into full matching. Let's go back to Python again and do the analysis. You can see if we call the propagate function, we can get a full matching that involves all cells. So let's run the propagation analysis. Okay, we should have the full matching for all the cells now. You probably remember we did the filter better matches analysis for the matched pairs. Because the matched pairs only capture a subset of cells, but now we propagate to the all the cells. So we need to run the filter better matches function again to filter away unreliable matches from the propagation for all the cells. So here we set the target as propagated. And here we use the filter proportion as 0.3. You can see for the Maxfuse package, because the weak link between the CODEX data and the RNA-seq data, this value was suggested to be set around 0.1 to 0.4. So here we use 0.3 to filter with the unreliable matched pairs. Let's run the analysis. Okay, we finished the filtration. So we should have full matching. You can see after filtering, we have 125,238 cells from the full dataset. So I'm going to stop from here for today's video. In my next video, we can extract the data for full matching, then perform the final joint embedding. So I hope to see you in my next video tutorial.